So let's now discuss position weight matrices, a more practical application of entropy and relative entropy towards identifying TF binding sites.
So now that we have some basic understanding of what entropy and relative entropy is, it's time to go back to discussing how to represent binding sites for a particular transcription factor that you've identified from, for example, the peak calling at the very end of the ChIP-seq protocol. And so suppose that you have a set of aligned binding sites of your given transcription factor. There's a couple of different common ways that TFBS are represented. So the classic way that was first used is basically what's called the consensus sequence, which is basically just a representation that summarizes the most frequently observed bases at each given position in the binding site. And so you'll notice for the consensus sequence representation, there's a whole bunch of letters that you probably don't recognize, like VDR, TWR, so on and so on. What these are basically new characters that are obviously not real bases, but they correspond to the occurrence of more than one base that is commonly observed at a given position. Basically, IUPAC came up with a consistent set of characters that are used to represent different combinations of bases at a given position. But the consensus sequence approach basically is not really used that frequently nowadays because everybody kind of recognizes that transcription factors can have different levels of sequence specificity at a given position in the alignment. And so, the precise specificity can't really be captured by consensus sequences. And so they were kind of replaced by what we'll discuss next, which is basically the sequence logo and position weight matrix. And so it's basically a precursor to the position weight matrix. What you can do instead of just a simple consensus sequence is that you can look at your alignment of your binding sites. And for every position in the alignment, you can basically calculate the number of times you see a given base at that position. And so that counting basically gives what's called a position frequency matrix, where the rows represent different bases and the columns represent different positions of the alignment. And so you can see that it basically just gives you a quick idea about which base is found, which position in the alignment. And this position frequency matrix is what's used to generate the famous sequence logo. And so the sequence logo, again, is a diagram, a visualization of the position frequency matrix, where columns in the sequence logo correspond to columns in the position frequency matrix. And the total height of a given column represents what's formally called as the information content of that position. And so for the purposes of this class, you can think about information content as basically what we refer to as the absolute entropy calculation. So basically, the height of the column is roughly proportional to how non-random the bases that you observe in that given position of the alignment is. And so the larger that number is, the more strongly the frequencies deviate from equal representation of all bases at that position in the alignment. The height of the individual letters basically, in some sense, corresponds to the contribution of each base to that total information content. So basically, the more a letter, a particular base, deviates from 25% frequency, the higher that column will be, the higher that base will be in the sequence logo in the corresponding position. And so sequence logos are good for visualizing a position frequency matrix and for giving some indication as to what the sequence specificity of the TF is at a given position in the alignment. But actually, more people would use what's called a position weight matrix to actually go and search new DNA sequences to find binding sites of a particular TF. And so the goal of a PWM is to be able to look at a short DNA sequence of length, the same length as the transcription factor binding site. And the goal of the PWM is to basically score that sequence for how likely it is to be bound by your TF of interest. So big positive scores correspond to high likelihood of a TF binding that sequence, and small numbers mean that TF is less likely to bind that particular sequence. And so in the middle of this slide, I basically show you how to go from a PFM, or position frequency matrix, to a PWM. And so you can see that a PWM is represented by a table of the same size as a PFM. And basically, the expression in the middle of the slide, log of PBI over PB, is how you go from the PFM to the PWM. And so at this point, I want you to recall our earlier slides about relative entropy. If you remember, relative entropy is basically a calculation about which computes how different the distributions P1 and P2 are from each other. And as you recall at the bottom of the slide, one of the key components of that relative entropy calculation is this log ratio. It was the log of the probability of seeing a character in the control fragments versus log probability of seeing a base in the bound fragments. And it's the negative of that. And so if you look at how PFMs are used to calculate PWMs, basically for a given position and base in the binding site. So for example, if you looked at, let's say, in column 2, if you look at C, basically C is observed, if you look at the position frequency matrix, C is observed 2 out of 5 of the input sequences. And so it's represented, so C represents about 40% of the input sequences at that particular position. And so how you go from, how you calculate the contribution of observing a C at the second position in the sequence is by computing the log of the probability of seeing a C in the second position based on your PFM. So that would be like 0.4 corresponding to 40%. And you divide that 40% by the probability of seeing a C in your control fragments. And then you take the log of that. And so basically that logarithm, that log ratio, is again going to be positive when you see that base more often in your PFM, so in your bound fragments, compared to your control fragments represented by PB. And the bigger the number is, the more positive that number is, the kind of the more deviating your base, the more frequently you see the base in that particular position compared to the control fragments. And so repeating that calculation, you can kind of fill out this PWM table and basically compute a score for each base in each position. And then basically what you do is that once you have a new sequence like TGC, TG, what you can do is you can basically, for every base in your input sequence, TGC, TG, you can look up the corresponding value that that base contributes to that position of the binding site. So basically, if you see the yellow entries highlighted in PWM, that corresponds to the sequence TGC, TG. And when you add up those values that are highlighted in yellow, you basically get the score of that sequence. And so in this case, it's 0.9. And so there's like a slight preference for binding TGC, TG compared to background expectation. And so the idea of detecting binding sites, so suppose you have like a genome, an entire genome that you want to scan for binding sites corresponding to your TF of interest. What you can do is once you have your PWM, you can basically slide a so-called window. So imagine having a window that's the same size as your TF binding site, or the same number of columns as your position weight matrix. And basically as you're sliding this window across the genome, you can basically calculate what the score of your TF is in terms of binding to that particular window of the genome sequence. And then you can basically compute scores, and you can score each window as you move along, or scan the genome in terms of, trying to find regions of the genome that have high score. And so typically speaking, when you're scanning a genome sequence for locations of a binding site, you can either look at the absolute score, which is basically what I discussed on the previous slide, where you just kind of add up the contribution of each base to the total score of a window, and that gives you your absolute score. Or what sometimes people do is they compute what's called a relative score. And so for any given PWM, you can easily identify the sequence that will give you the highest possible score, right? And so you can do that by basically looking at each column of your PWM, just checking out which base gives you the highest score, and just making a fake sequence, for example, that has that base at that position. And after you look at all the columns, you'll basically arrive at the sequence that gives you the highest score from the PWM. In a similar way, you can also calculate what the smallest score you could get from a PWM is by looking at the smallest number in every column and just adding those up, and that'll give you a minimum score. And so given a maximum score possible and a minimum score possible, as well as your absolute score that you get from a given sequence, you can use the equation on the bottom right to calculate what's called a relative score, which tells you how, what percent of the maximum possible value does the sequence of, that is contained within a window give you relative to the maximum and minimum that you could have gotten from the position weight matrix. And so in practice, there's a lot of people, a lot of people who spend a lot of time basically building software to, number one, kind of look through the literature, look through ChIP-seq data, through SELECT experiments, and build up libraries of sequence logos, as well as position weight matrices for different transcription factors from different species, and they basically create these big databases of logos and PWMs. And so on the left, I've given you some examples of some of the most common databases that people use for if they want to download like a PWM for a transcription factor of interest and then use it to scan their own genome to figure out where TF binds.